Therapeutic approaches in epigenetics are now being translated into clinical practice. The epigenome is no longer just a research concept, as several epigenetic drugs have been approved by the FDA, marking significant progress and pointing toward future therapeutic developments. Epigenetic drugs aim to reprogram cells by reversing abnormal epigenetic marks, particularly in cancer, where tumor suppressor genes are frequently silenced. One major approach involves DNA methyltransferase inhibitors, such as azacitidine, which act as nucleoside analogs. These drugs incorporate into DNA during replication and trap the DNA methyltransferase enzyme, leading to a global reduction in DNA methylation. This allows previously silenced tumor suppressor genes to be re-expressed. Another approach is the use of histone destyles inhibitors, which block enzymes responsible for removing acetyl groups from histone by maintaining histones in an open, acetylated state. These drugs promote transcription of genes that induce cell cycle arrest and apoptosis, or programmed cell death. Currently, the FDA has approved azacitidine and decitabine for the treatment of myelodysplastic syndromes and acute myeloid leukemia, reflecting their clinical relevance in hematological malignancies. More recently, in 2024, revumenib was approved for specific types of relapsed leukemia, highlighting the expanding role of epigenetic therapies in oncology. A second major focus is gene therapy and CRISPR technology. CRISPR-Cas9 uses a guide RNA to direct the Cas9 enzyme to a specific DNA sequence, where it creates a double-stranded break, allowing deletion or correction of a mutated gene. In contrast, epigenetic editing uses a catalytically inactive dead Cas9 that cannot cut DNA. This dead Cas9 is fused to epigenetic writers or erasers, such as DNA methyltransferases or histone acetyltransferase, enabling targeted activation or repression of genes without altering the underlying DNA sequence. In December 2023, the first CRISPR-based gene therapy was approved by the FDA for sickle cell disease, marking a major milestone in genome-targeted therapies. In addition, ongoing clinical trials are using CRISPR to disable PD-1 in T cells, enhancing the immune system's ability to fight cancers such as lung cancer. Looking ahead, the future of epigenetic therapy lies in addressing several key challenges. These include combination therapies, where epigenetic drugs are used to prime tumors and increase their sensitivity to immunotherapy or conventional chemotherapy. Another major goal is the development of epigenetic drugs that target specific cell types, reducing the side effects associated with global epigenetic changes and aligning with precision medicine strategies. There are also important ethical considerations. These include the risk of off-target CRISPR edits, which could lead to unintended health consequences or secondary malignancies. Germline versus somatic editing remains a highly controversial issue, particularly the possibility of editing embryos, which would permanently alter DNA across future generations. Finally, there are significant cost implications, as these advanced therapies may currently be accessible only to wealthier populations, raising concerns about equity in access to life-saving genetic and epigenetic treatments.